So chapter 6 is called the cellular basis for Mendelian genetics. And what they actually mean by that is cell division. So we're going to go through the two different types of cell divisions, mitosis and meiosis, and then we're also going to extend that into producing sperm and eggs. And then we're going to talk a little bit about some of the errors that might be produced during the process of those gamete cells. So after Mendel did all of his work, it was essentially lost for about 35 years. And it was lost until cytologists or cell biologists, those individuals that are studying the cytoplasm, that's where we get cyto, until they discovered it. And they discovered it because microscopy improved. Biologists began to look at cells in more detail and to observe the processes that were happening in them. When they started to look at those cells, they began to see that there was a link between the way that chromosomes moved during cell division and Mendel's traits. So there was a link between the passage of that information and the actual information encoded on those chromosomes. So we're going to start by talking about the cell cycle. So in a eukaryotic cell, and actually in prokaryotic cells as well, but we're mostly going to focus on eukaryotic cells today, there are a set of phases that the cell must go through throughout its lifetime. So basically there's growth, there is development, there's performing the job, and then there's cell division. So a bacterial cell, such as an E. coli, can divide as quickly as every hour. So you get very quick multiplication of cells, very fast cell division. Eukaryotic cells, on average, reproduce once every day. So if we look at this cell cycle on a 24-hour period, on a 24-hour clock, the process of cell division is going to take about 1 hour. So that process of cell division is actually really quick compared to the rest of the lifespan of the cell. Most of the cell cycle is spent in what we call the G phases. Those are the growth phases. So there are two major parts of the cell life cycle. One is interphase, and interphase simply means the time between cell divisions. So interphase is going to go from this point where we start at the end of cell division. So we start with a new cell and we go through this process of growth, DNA synthesis, and more growth before we get back to cell division. In the G1 phase, this first growth phase, we see the cell performing sort of normal cellular functions. The cell is growing, so the physical size is growing, it's expanding, it's producing proteins, it's doing its job within the organism. Now, at some point in this growth phase, the cell has to commit to continuing on into the process of cell division. Now, as it readies for the process of cell division, it's going to enter the S phase, or the DNA synthesis phase. Now, this is where that process of DNA replication happens that we talked about earlier in the semester in chapter 2. Once DNA synthesis occurs, the cell enters a second growth phase. Now, this growth phase is slightly different. It's really characterized by preparation for mitosis. So the cell checks to make sure that there's no major problems in the DNA, all of the repairs have been made, any breaks that might have happened have been repaired. And at that point, at the end of G2, the cell is going to evaluate and say, yes, we are ready for cell division. And that is when we enter the process of mitosis, which is cell division for somatic cells. So somatic cells are basically all the cells in our body that are not gametes. That is to say, all the cells that are not sperm or egg. So this is our skin cells, production of red blood cells, epithelial cells, the cells that line the inside of your stomach, things like that. Some cells actually also have a phase called G0. cells that are in G0 actually don't divide. So our nerve cells don't divide. Once they're produced, they don't divide. Liver cells don't divide unless the liver is damaged. So some cells, instead of entering G1 and then committing to continued growth and cell division, actually enter this G0 phase where they remain sometimes for the lifetime of the organism. So before we move on, I want to review a couple of terms, and these are chromosome terms that we learned in chapter 3. So recall that in eukaryotes, they often have multiple copies of linear chromosomes. So each individual is going to get a set of chromosomes from mom and from dad. And because those chromosomes in the sperm and the egg combine, the sperm and the egg each have half the number of chromosomes. Now, there are some other organisms that are haploid, but in general, we're going to be talking about gametes as being haploid. That means they have one copy of each type of chromosome. And our haploid cells are going to be produced by the process of meiosis, and they're going to be produced in the gonads. So the gonads are the organisms, the organs in the organism that produce the sperm or the eggs. So let's say that we have a cell and it is haploid. It's got 3 different types of chromosomes here. We can see they are physically different. I've drawn condensed metaphase chromosomes. That's why they look sort of short and fat. But we'll get back to metaphase in a few minutes. So here we have one copy of 3 different types, type 1, type 2, and type 3. So here our single copy of each type is going to be equal to 3. So we have 3 chromosomes. We have one copy each. Now cells in eukaryotes tend to be diploid. That means we have 2 copies of each type, one from mom and one from dad. All right, so these are going to be produced, diploid cells are going to be produced from the fertilization of the sperm with the egg. And then that cell is going to continue to divide and produce somatic cells. So remember, those are our non-gametes, our non-sperm or non-eggs. And they are going to be produced by the process of mitosis. So if we were to draw this same cell as a diploid, if we had our egg, that was n equals 3, and our sperm, that was n equals 3, and we produced a fertilized gamete or a zygote, we would end up with a cell that is 2n equals 6, because each of those gametes is going to contribute 3 chromosomes. Now, let's say that dad contributes these blue chromosomes, and then mom is going to contribute some red chromosomes. Now we can see that they contribute the same type of chromosome. And we have 2 copies of each type. So here we have type 1, here we have type 2, and here we have type 3, but we have 2 copies. So we have total of 6 chromosomes, 2 copies of each type. So let's look at this cell again. I've rearranged the chromosomes here. I've put them near one another so we can see the different chromosomes that we have. So remember, this was our haploid cell from the previous, our diploid cell from the previous slide. Haploid number of n equals 3. A diploid number of 2n equals 6. So this is our diploid cell here. We have 2 copies of each type. Now these type 1, type 2, and type 3 are homologous chromosomes. We might also call them homologs. So homologous chromosomes are going to have the same physical characteristics. So their centromeres are going to be in the same place, their arms are going to be the same lengths, and they are going to contain the same genes at the same locations, and that's really important. so same genes at the same location. Now, that's not to say that they are completely identical, because they might differ in alleles. So they might differ in versions of the genes that they have. So you might have one parent that is brown-eyed and one parent is blue-eyed. That eye color gene is going to be in the same location on each homologous chromosome, but they contain slightly different information. Now, when we go through the process of DNA synthesis or DNA replication, that S phase, we are going to produce sister chromatids. So sister chromatids are going to be essentially identical all the way along because they are produced by DNA replication. Now, because of errors in DNA replication, they might differ by a few base pairs, say 10 base pairs out of several million, but they are still going to contain the same alleles at the same location. So we are going to draw these as paired chromosomes, and we are drawing them still connected because they are connected because of DNA replication. So now, where we had homologous chromosomes, we now have homologous chromosomes that are ready for cell division. The chromosomes have been copied. They're still connected, which is how we know that they're identical. And each of these are going to be called sister chromatids. Here's another pair of sister chromatids, and here and here. So these chromosomes that are connected at the centromere and produced by DNA replication are your sister chromatids. Now notice that I really haven't drawn the homologous chromosomes touching one another because they really don't associate with one another. They never sort of come together except in one very specific instance that we'll talk about in a few minutes. It is only sister chromatids that are physically joined most of the time. So let's look at a karyotype. Remember that a karyotype is essentially an image of chromosomes. Now these are our super condensed chromosomes. They are called metaphase chromosomes. We'll see what we mean by metaphase chromosomes in just a minute. But from the karyotype, we can see this 2N or diploid situation. So this individual, I believe this is a rabbit. Can't remember. I believe this is a rabbit. If we look, we can see that we have 2 copies of each type. So we know that this is diploid. This is a 2N organism. And we can see that we have 19 regular chromosomes plus sex chromosomes. Now, if we were to count these up, we would find that there are 40 chromosomes altogether. 2 of each kind up to 19, and then 1 each X and Y, so 2 sex chromosomes. They count as one type. We can also look at this and determine that if we were to look at the gametes and look at the haploid cells, we would find that N equals 20 because there are 20 different types, 19 autosomes and 1 sex chromosome. So we have 19 autosomes or non-sex chromosomes and 1 sex chromosome in our diploid situation. So each one of these little chromosomes here is going to be a sister chromatid. So here we see our sister chromatids that are joined together after DNA replication. So sister chromatids are found here in these highly condensed chromosomes. And then in our karyotype, we have our homologous chromosomes. So each of these is a set of homologous chromosomes. All right, so let's look at another karyotype. So this is showing 1, 2, 3, 4, 5, 6. We can keep counting. 23 types of chromosomes. But look at this one. There's only one copy of each type here. Now these are our sister chromatids, just like we had up here, but we only have one type. So this cell is actually a haploid cell where N equals 23. This is an organism called C. tigris, which is actually a snail, and it's just by coincidence that it happens to also be N equals 23. But here, just to show you how they differ, is our haploid karyotype for this organism. All right, so just a quick review. Sister chromatids are produced by DNA replication or DNA synthesis. They should be essentially identical. Now, DNA polymerase, as we saw in previous chapters, is not foolproof. It does make some errors. So there might be a few errors difference. Chromosome 1, they estimate, has about 10 base pairs difference between sister chromatids. But for our purposes, we're going to basically say that sister chromatids are identical. They're like identical twins. And then if we look at the homologue, the other one, if one came from one parent and this one came from the other, they're going to be slightly more different because remember, they contain the same genes, but they might have different alleles. And so the differences between two homologous chromosomes tend to be a bit bigger. In this case, they're estimating that two homologous chromosomes, chromosome 1s, are going to be about 250,000 base pair differences. So you can see why I'm essentially calling sister chromatids identical, because they are far more similar than they are to the other homologous pair. All right, so let's dive into cell division. So we are going to talk first about mitosis, which is the production of somatic cells. And in mitosis, we are going to separate sister chromatids. So if we go back here, in mitosis, we are going to pull our sister chromatids apart. Okay, we're going to separate those guys. Now, the outcome of mitosis is that we produce a diploid cell. So we start with a diploid cell and we produce a diploid cell. So this makes sense because if you were to think about producing a somatic cell, let's say a skin cell, that skin cell still needs to be a skin cell. And so it has to have all the DNA that it started out with. And so if you start out with 2 copies of each type of 23 chromosomes or 46 chromosomes, you need to end with 46 chromosomes because those cells have to immediately start doing their job as being a skin cell. So let's talk about these steps in mitosis. I'm not going to go into extreme detail because you probably had this in Biology 101. So this is going to be a bit of a review. If you have questions about the specific stages, please let me know. But this image pretty much has everything you need to know. So just very generally, we're going to start at the S phase in preparation for mitosis. Remember, this is interphase, or the time between cell divisions. So during the S phase, we are going to copy our chromosomes. So here, let's start with our chromosomes. We have 3 different types here for 6 chromosomes. So in this case, our cell is 2n equals 6. Now, the blue chromosomes are one type. The the orange, red are one type, and then these purple, blue are another type. So during that S phase, we are going to copy our DNA, and we are going to produce sister chromatids. So here we have our sister chromatids here. You can see that by the X shape that they make. So we have sister chromatids for all of our homologous chromosomes. So this set of blue are the homologous, and then we've got the red and orange that are homologous, and then those purple ones that are homologous. So during the process of mitosis, we are first going to condense our chromosomes. That condensing actually starts in the very end of G2, which is very short, those chromosomes begin to condense, and then they are going to line up along the center of the cell. They're going to be pulled apart, and then the cytoplasm of the cell is going to be separated to produce two daughter cells. So if we look at prophase, in prophase, we finish up condensing our chromosomes. So whereas we started out with this jumble of spaghetti chromosomes over here, we have these very tightly condensed chromosomes. We start to produce microtubules. These are structures in the cells that are going to bind to those chromosomes, and they're going to actually do the work of lining them up and separating them. So our microtubules are going to grow towards our chromosome. They're going to attach at a location on the centromere called a kinetochore. And then they are going to continue in sort of a push-pull motion until all of those chromosomes line up in the center of the cell at what we call the metaphase plate. So the metaphase plate is sort of the equator of the cell. Now they have to line up this way because they need to be partitioned equally into the cell. Now that partitioning happens during anaphase. In anaphase, those microtubules begin to shorten, and when they shorten, they pull apart our sister chromatids. So these chromosomes here were originally bound together after they were produced via DNA replication, and then they were pulled apart by the movement of those microtubules. So we've pulled those chromosomes apart, but you'll notice that we still have homologous chromosomes. So those sister chromatids of each homologous pair are pulled apart, but we can see that there is one half of each of those sister chromatids going towards the pole there. So then we continue on in mitosis. In telophase, we start to see the cytoplasm start to pinch together. That's actually called cytokinesis. And that process sort of happens at the same time as telophase. In telophase, those chromosomes sort of come back together. The nucleus reforms. So back here in prophase, the nucleus actually broke down. The nuclear envelope actually broke down, and it begins to reform in telophase. Cytokinesis continues to occur to split that cytoplasm and to partition all the organelles within the cell. And then once telophase and mitosis is over, those chromosomes decondense. The nucleus is fully formed, the cell membrane is fully pinched, or if it's a plant, a new cell wall is formed in between. So the end result is that we maintain that diploid cell. So we still have 2n equals 6. And if you count them, 1, 2, 3, 4, 5, 6, there are 6 chromosomes in there with 2 copies of each type. So we have maintained diploidy. So we've maintained the diploid nucleus, but we also have produced genetically identical cells. Okay, each cell has an identical copy of the chromosome that was in the parents, and each cell has 2 copies of each type. So we have maintained, we should have an exactly identical cell to the parent cell. Now keep in mind, if this is a skin cell, you want it to be that way. You want that skin cell to continue to produce skin cells, and so it needs to be genetically similar. Prophase and metaphase tend to be the longest. When these chromosomes are condensing, those microtubules are forming. And then anaphase, once everything's lined up, anaphase is actually really short. It's the shortest part of mitosis. But keep in mind that this whole process of mitosis really only lasts about 1 hour if we're looking at a 24-hour time scale. So as part of the process of mitosis, we need to form a couple of structures. So going on outside of the nucleus, the cell is actually dividing or replicating depending on the organelles, sorting the organelles and getting ready to move them as well. But we're in genetics, so we're going to worry just about what's happening in the nucleus. Just keep in mind that those other organelles are moving as well. But all of this process is regulated by checkpoints, and we'll talk about those in a minute. But one of the things that has to happen before mitosis can occur is the production of the centrosome. Sorry, this should actually say centrosome, not centromere, centrosome formation. And the centrosome is really important because the microtubules, or the fibers that are actually going to attach to those chromosomes, grow out of the centrosome. So centrosome production begins in G1 phase. A centrosome is actually a pair of two structures called centrioles. So two centrioles make up a centrosome pair. Those centrioles are going to be duplicated when the DNA is duplicated. And then in G2, they're going to be, in preparation for mitosis, they're going to begin to move toward the poles. Now, the reason is that they are going to be the structures that are actually moving and pulling those microtubules. So the centrosomes have their own duplication cycle. They're replicated just like DNA, and they're going to be separated just like the DNA in the cells. So once those centrioles are duplicated, they will begin to start to produce microtubules, or we might also call those spindle fibers. The structure, this early production of those spindle fibers, is called an aster. So aster means star. So it's going to produce this star-like formation. That star is produced because microtubules are going to grow out of those centrioles and the centriole pairs that produce the centrosome. So we've got a different image here. This is our centrosome. We've got one half of the centrosome on each side. So each of those is a centriole. So remember, two centrioles makes up a centrosome. The microtubules are going to grow out of the centrosome. They are going to grow toward the chromosomes, and eventually they are going to attach to the centromere of those chromosomes. Now, microtubules are made up of repeating subunits called tubulin. There are two different types. There's alpha tubulin and beta tubulin. And you'll see that they are alternating. So one end of this tubulin chain is going to be attached to the centriole. It's going to grow by the addition of tubulin subunits, or it's going to shrink. We'll see them shrink also by the removal of those tubulin subunits. So microtubules are going to grow out of those centrosomes. They are going to grow towards the chromatids, and they are going to attach to the kinetochore, which is a specific location at that centromere, sorry. Remember, the centromere is that little pinch in the chromosome. It's a really highly condensed region, and it is one of the structural aspects of a chromosome. So those microtubules are going to grow towards your sister chromatids. Some of them are not going to attach to chromatids. They're actually going to attach to one another. And those ones that are going to attach to one another are going to be performing the function of stretching out the cell. So they're going to attach to one another, and then they're going to sort of stretch apart. And that is going to elongate the cell. Now that allows for room for that cytoplasm to be pinched off in the middle. So overall microtubule functions, they're going to attach to and move chromosomes. They're going to elongate the cell. And then their third function is to sort of pinch the cell membrane during cytokinesis to pinch the cytoplasm apart. So they've got a lot of important functions here. So I forgot about my little animation. I forgot to click it. So this is just saying that your microtubule is going to connect at your kinetochore of each side of the sister chromatid, each sister chromatid.